Anil Diwan, president of Nano Viricides, is going to tell us how the company is combating viruses in a small yet mighty way. Thanks for joining me, Anil.
Thank you very much. Thank you for the invitation.
So when I think of nano, I think of small. What are nano viricides?
So nanohericides are exactly that. They are very small micellar particles. They are very flexible. They are like, think of soap, but think of all the soap molecules connected to each other in a chain. That's what nanohericide is like. And what we design here is a machine, a full nano machine that can just grab the virus and destroy it. Imagine that in the stream, in the bloodstream, in the lymph nodes.
Incredible. What viruses does it target?
We have done a lot of viruses already. So we have targeted influenza, we have targeted HIV, we have done work against dengue, Ebola, to name a few. We are now focusing on herpes viruses. It's a ubiquitous virus that is very HSV1, HSV2, VZV. These are ubiquitous. They are almost everywhere in everybody, and they are a major threat. It's just not being talked about as much, and it's an important problem.
Absolutely. And you say it's in the form of a machine. Does it change within different viruses?
Yes, there is one small part that we have to change based on the virus. So think of it this way. The virus is a body, it's a particle that lands onto the cell surface. It binds to the same point on the cell surface no matter how much it changes. Viruses mutate all the time, but they bind to the same thing. Now, so what we try to do is we try to copy that same thing where the virus is going to bind to, and we make a matrix, a surface, where it will look like the cell surface to the virus from the outside. So the virus will come and seek the nanoviricide and bind to it. When that happens, the nanoviricide actually opens up and engulfs the virus, sort of like a Venus flytrap.
That's a really great analogy. And how is this approach better than what is out there right now?
So there are two parts to how virus grows. Viruses are semi-living beings. They don't grow by themselves like bacteria do. They have to go inside the cell, open themselves up, and then grow. In order to do that, they first have to go inside the cell. And then from the inside the cell, after they have grown there, they come out and then they try to infect other cells. So what we do is we intercept that ability of the virus from attacking a new cell. And the existing approaches that are there, most of them other than vaccines and antibodies, all the other approaches are about interfering with the virus growing inside the cell. And most of those approaches, fortunately or unfortunately, they tend to interfere with human host machinery. And when they do that, automatically they become toxic chemicals. So ours is an approach that is non-toxic. It stays outside the cell most of the time, and it engulfs the virus completely and destroys it completely.
What results have you seen in your clinical studies?
We haven't gone clinical yet. We will be in clinical trials hopefully by the end of this year.
That's so exciting. So what kind of news do we look forward to?
Well, we hope will be great news that we are entering the clinical trials, but we have de-risked the clinical trials already. So what we are doing now is we are working against herpes class of viruses, HSV1, HSV2, and VZV, shingles. So shingles, I think everybody knows it. When you reach 45, 55, that age, you will get that big rash for 3 weeks or so. It's very painful rash. That's the one that we are going to use as a first indication. And the reason for that is we can develop a skin cream for it, and that gives us a much earlier regulatory pathway to go through. Much quicker regulatory pathway to go through towards clinical trials approval. The other reason we chose shingles is that there is no animal model for shingles. Shingles is a particular virus that only infects human beings. So for that to show efficacy, we were fortunate to be able to introduce to a lady, Dr. Jennifer Moffat at SUNY Syracuse, who has developed a human skin-based model. Now that means we are infecting human skin with the shingles virus and then applying our cream to that and see whether it is effective or not. So that means we are completely de-risked from the human clinical studies. Of course, there can be some surprises, but we don't believe that there will be any. So far, all the drugs that we have, many different drug candidates that we have in the pipeline, all of them have showed tremendous safety. The shingles candidate has gone through non-GLP studies already towards IND, investigational new drug application, and it has been very safe. It is now into the GLP part, the regulatory part, where the I's are dotted and T's are crossed, that part of the study. And there also we have seen that it is fairly good. it is non-toxic, completely non-toxic. So from safety point of view, we don't expect any problems. From efficacy, like I said, we have already worked on human skin itself. So we don't expect any problems.
That is some really exciting, unique, and interesting news, and it will be very helpful. Good luck and congratulations.
Thank you very much.